Good morning, everyone. He's in France. This is the afternoon here, the end of the day, actually, because it's winter. So I'm very pleased to share these slides with you. And I urge you to don't hesitate to interrupt me if you have questions. I want this to be as interactive as possible, if you have the possibility to do it, of course. So the topic today is actually the result of a few years of research in my group, where the idea was, the question was, what can we do with SAXS, using SAXS with membrane proteins? And so we came up with a strategy that I'm going to develop in the next slides. And the strategy is based on the combination of two programs. Actually, one program is called Memprot, and the other program is called DADIMODO. So Memprot for membrane protein, and DADIMODO for domain docking, data-directed module docking. And so these two programs combined can give an answer on how to use SAXS with membrane proteins. So the work in my group was done primarily by these people. So Maciej Baranovski, who was a postdoc, Aurélien Thureau, who is a scientist on my Beamline, and Olga Rudenko, who is a specialist of, she's a mathematician, and she's a specialist of the kind of algorithm that the genetic algorithm that we are using for DADIMODO. So this is a picture of our Beamline here. And this picture was taken about 10 years ago, where there were only few beamlines in the hole. And what is nice with our synchrotron in particular is that we have the light from outside. So when you work, you are aware if it's day or night outside, and this is usually very good for our mental equilibrium. So the synchrotron is situated 30 kilometers to the south of Paris, and our Beamline was one of the first built Beamline in 2008, and from the beginning was shared in terms of scientific cases between structural biology and soft condensed matter. So it's a typical SAXS Beamline where we have a flux on the third generation synchrotron, classical flux of about 5 to the 12 photons per second at 12 keV. And the beam is relatively large, 400 microns times 100 microns, which is good for solution samples because the intensity, the flux is diluted on the whole sample, and so there is less radiation damage. The principle of the experimental hatch is the following. We have a big table here, which is motorized, and on this table we can put any kind of sample environment. So there is a lot of room, so we can change sample environments every week or even twice a week. So we do this in a few hours, so very little beam time is misused. And the detector is in this very large vessel here, which is entirely in vacuum, as you can see it from inside. You have the detector here at 6 meters from the entrance. And what is good with this geometry is that you can change the distance from the detector to the sample very easily. Actually, the user can do it just by clicking on the button, since everything is motorized inside. So here we have an example of a very classical capillary holder for chemistry, where the capillary holder is in air. So we have here the entrance of the vacuum of the detector chamber with a small window here. And we have the beam coming from the optics through this tube and another window here. So the sample in this case is in air in between two windows. But for biology, because the signal of the scattering of the solutions, or the scattering signal is very low, it is better to minimize as much as possible all parasitic signal, and in particular the parasitic signal from the windows. So for biology, we made a sample holder which is in vacuum, so we have a capillary through which the sample goes and flows, and the capillary is in vacuum. And as you may know, because you have now followed this workshop, it is important for proteins in solution to have a very monodisperse sample to be not, for the data not to be biased, to be confident in the data, and so to have models that can really reflect what the true data are. And so for this reason, we have this combination of HPLC size exclusion chromatography system with which we can purify the sample online. Okay, so here we have two different pumps, so which role is slightly different. One of the pump is used to really elute the sample into the capillary cell, while the other pump is used to equilibrate another column for the next experiment to come. In this way, there is no beam time loss when you change the buffer or when you change the column. In combination with these pumps, we have a UV detector, of course, to determine the concentration of as a function of the volume eluted. And we have the SAXS cell, and we also have these 18 angles MALS and DLS online, which can provide complementary information about the shape and also the size of the protein. And also very important for membrane proteins, we also have this refractometer here online, which can give also a concentration of molecules that would not absorb UV light, like, for example, detergents. So the motivation, which was my motivation actually, which started about 10 years ago, was what can I do to apply SAXS to membrane proteins? Because we know that SAXS is good at monitoring conformational changes. And of course, membrane proteins, as all proteins almost, they do undergo conformational changes when, for example, they are liganded or with an effector, so that their conformation can change, or when the pH changes, or when you have different molecules outside the membrane that can trigger a modification of the conformation of the membrane protein. So the question is, how can we use SAXS to monitor membrane proteins conformational changes? So this was the first questions. The main objective we wanted to reach, but before reaching this objective, we realized that there was a much simpler question that we still could not answer so easily, is how can we use SAXS with a membrane protein of already known structure? So it's already difficult to use SAXS to do something clever with SAXS with a membrane of known structure. So with a membrane protein of unknown structure, this is even more difficult. So we are going to go step by step. So our strategy is a two-step strategy. So we must first start with a construct of the membrane protein that is known from crystallography, for example. And from this construct here, we solubilize it in a detergent solution, and we produce a SAXS curve in some way that I will go into detail later. And from this SAXS curve, we construct a model for the corona of detergent that is around the known structure. Then, what we are interested in at the end is, if you have a construct of the same protein but with an additional domain, how would the conformation of the whole protein be? What would be this global conformation? Then for this, we use this program called DADIMODO, and that allows you to fit the curves, the SAXS curve, by using rigid body docking of known domains. But the nice thing is that DADIMODO includes the possibility to use the model of the corona on the known part of the protein. So this strategy was written in a book recently. And the guinea pig protein that we used now 10 years ago for this was aquaporin 0, which is a water transfer molecule that is present in the lens of mammal eyes, and it's the main protein of the lens. Actually, 60% of the membrane protein content. So it's natively a tetramer, and it appears that from the cortex to the core, the extra part here, these linkers are truncated, and so the inner part of the crystalline is made by this tetramer, but truncated tetramer. And it appears that the truncated tetramers, they crystallize as octamers, while the cortex, the full aquaporin, crystallizes as tetramers. So the initial goal of this study was to check whether in solution we really had these octamers present, existing, or if they were biased from the crystallization process. And actually we couldn't reach this goal, we couldn't answer this because we were not able to purify the truncated octamers, unfortunately. But in the way to this, we learned a lot about what I'm going to talk now. So we made from directly purifying from eyes of veals, we obtained a concentration of about 4 milligram per milliliter, and we obtained a lot of solution, 2 milliliter. And this kind of concentration is really what you need if you want to perform a nice SAXS experiment, if you have to use size exclusion chromatography online. So, as you know, when you solubilize a membrane protein in a detergent solution, then you have a mixture of your protein in complex with the detergent, so with the detergent corona, but you also have these free micelles of detergent. So there are two problems then for SAXS. First, you have a mixture here, so you know that SAXS is sensitive only to the average of the signal. So when you have a mixture, you have to find a way to separate the components. And then also you have this detergent belt, what to do with it. So the mixture problem is directly solved by using the size exclusion chromatography online. Since if you pre-equilibrate your column with a solution of detergent above the CMC, you will have these free micelles at a given concentration throughout the whole column. And when you are going to elute your sample, the proteins will elute faster than the free micelles, so they will travel along the column environment where the concentration of free micelles will be constant because the column was equilibrated. So at the end, if you have this elution peak here and you collect the SAXS data under the elution peak and you subtract the data collected on the buffer, then you will end up with scattering curves from pure protein complexes. So this is symbolized here, where you have the peak here from the mixture of the free micelles from the buffer and the corona, the complex of protein with the detergent corona. And the free micelles that were in excess in the injected solution, they travel slower in the column, so they will be eluted afterwards. But this can only be seen using the refractometer because they don't absorb UV light. So this is why also it is important to have this refractometer to be sure that the two peaks here, the peak of protein and the peak of free micelles, are eluted separately. In addition, if you compare the signal from the refractometry at the peak of the proteins and the signal from the UV absorption, and if you know the dN over dC of the protein, which is the equivalent for refractometry as the contrast is for SAXS, so this is usually tabulated. If you know the dN of the dC of the detergent, which is also tabulated, and the epsilon of the protein, then you end up with the possibility to calculate already the number of detergent molecules around your protein. And this is what we get here from using this comparison of UV and OD, 240. Then from the peak here, you can collect, we have collected the SAXS data, of course, and we have produced here the elution profile of, in blue, I0 over the concentration of the protein, so normalized I0, in green, the RG, and in red, you have the pure I0, and in dark, the dark line here is the UV absorption. So you can see that there is a perfect superposition of I0 over OD, which is, whose consequence is that the blue line is very flat. So that means actually that there is no depletion of detergent throughout the peak. So we truly have a monodispersed solution here. And so if you have previously calibrated the beamline using water, for example, you are able to produce the I0 value in absolute units. And if you are able to do that, and because you know the concentration from OD, then you are able to determine the mass of everything there. And so comparing the SAXS I0 and the UV, so without refractometry this way, you can also determine independently the number of detergent molecules. And we ended up with something very similar to what we had before by comparing UV to refractometry. So we were happy about that because all the data were coherent in between them. So then we were confident that the final curve that we produced by using the average of the curves under the peak here was truly the curve of the complex protein detergent. So here is the average that we get. And so the question now is how to find a model which fits the curve. So despite we have some ideas about the how the protein looks like, we could just simply start by using an ab initio method, like the program DAMIN or DAMIF, for example, or Monza. But we cannot do that because we have several different electronic densities here. And the DAMIF or DAMIN, for example, they rely on the assumption that the density of the whole object is constant. And if you don't have a second curve with, for example, different contrasts like what you can obtain from SANS, you cannot use Monza. So we cannot use these simple ab initio methods. Instead, that was our idea. Because we know the structure of the protein, let's concentrate on modeling the corona of the detergent. And let's do it in a geometrical way. So let's combine the PDB structure of the protein with some coarse grain modeling of the corona of detergent. And we could model this using a torus whose parameters are described here. And so this is the main idea that we had. So the corona, we make a PDB file where the protein is described by its atoms, and the detergent corona is also described by atoms, but these atoms actually are only there to fill a volume, a geometrical volume. And you see here that we use two kinds of different atoms from the table of chrysol. I guess you have heard about chrysol by now. So the light gray atoms are there to mimic the hydrophobic part of the corona, and the dark gray atoms are there to mimic the hydrophilic part of the corona. And we have selected those atoms and the grid also, so the distance between the beads that fill the volume, in such a way that the resulting density really corresponds to the known density of hydrophobic tails and of the hydrophilic heads of the detergent. So does it work? So to know if this works, we checked the consistency of the approach. And for this, we model a core shell ellipsoid, which is known to be analytically solvable. So we can produce an analytical curve from this. And so the idea here was to compare a curve directly from the PDB model made with the strategy we just proposed, and a curve made with and the fitting curve, and the curve made analytically with the same parameters. So we constructed this PDB file with these beads here. We calculated the curve using chrysol, and we tried to fit it using the analytical function using the program SAXSFit. And this is what we got. So we have the dots represent the curve from the PDB file, and the red line is the fit from SAXSFit. And the fit gave the following parameters, which are very close to the initial parameters that we put there. So of course they are not identical because the beads are not so small, so there is a kind of a roughness here which is not accounted by the model, but still we are very close. So also the densities, the ratio between the two densities also was imposed in the model, and we can see that the densities as well as the geometry are well reproduced by the analytical curve. This way we validate, this is the way how we validated our approach. So we computed the best possible torus to fit the data, and this is what we got here. So the experimental data of the complex, and in dark, in black, the black dots, and the fitting model using the best possible torus here gives the blue curve here. So as you can see, we are already not so bad. So we can reproduce the position of the two maxima here and the position of the minima. And this only with three free geometric parameters, which were the total thickness of the corona, its width, and the thickness here, T, of the hydrophilic part. But we were not completely satisfied by the quality of the fit, so we added a parameter in the model, and that parameter is ellipticity here. So instead of being a circle here from the Z projection, it's now an ellipse. And now, as you can see, the experimental data are very nicely fitted. So, of course, this ellipticity represents a kind of average of the fluctuation of the shape of the corona. It's not something that you can really believe, but it's good because now we have a model that can be used as a base for further fitting, as I explained in the two-step strategy. In addition, from the number of beads here, you can count the number of beads, so you can count the volume that you get for the hydrophobic tails and the hydrophilic heads, and dividing the global volume here by the volume of a single detergent molecule, so you can end up with a number of detergent molecules. And we are, again, very close to what we got with the previous completely independent methods of determining the number of detergent molecules. So this was published 8 years ago. And since we made a program that helps users that would be interested in using the same approach, and the program Memprot is simply a program that will scan on a step-by-step all the parameters, so it's relatively long and very simple in its algorithm. It will scan all the parameters of the model, A, B, T, and E, and even now we have implemented another parameter, which is phi, which is the orientation of the ellipse with regard to the projection of the protein. And the Memprot computes the chi values using Chrysol for each of the model generated this way. And at the end of the program, you have these correlation maps here that compare each parameter with another one, and compare in which way it plots the chi values as a function of the two parameters. So you can see that some of these couples of parameters do result in a kind of single minimum here, but two of them in particular, B and A, are anti-correlated, meaning that you may find the same kind of agreement of the fitting curve if you increase slightly A and simultaneously decrease slightly B. And what are these two parameters? A is the thickness of the corona, and B, it's its width. So by decreasing the thickness and increasing the width, you're just conserving the number of molecules. You're just conserving the volume. So it is not that surprising that within a limited range, if you have the same volume, even if the thickness is slightly smaller and the width slightly larger, you will end up with a similar fit agreement. So this reminds us that with SAXS, as usually, there is never a single model that will fit the data. So we have to live with this. But these correlation maps are also useful in the way Memprot should be used because, of course, you're not going to start to launch Memprot with a very small step for each of the parameters blindly. Otherwise, it can last for days or months of calculations. So you will start with very loose steps first, and kind of zoom then on the minimum area. So starting from 28 to 32 angstroms for A, for example, with only 1 angstrom step, and then zoom into a smaller area here where you will relaunch Memprot using a thinner step. So this is exemplified here, where we made this strategy of thinner and thinner steps. So first, the first fit was, as I said, using steps of 1 angstrom. And the best model that we got corresponds to the red curve here. And you can see that, okay, the experimental data are in blue in this case, and the first fitting curve is the red one here. So you can see that the minimum position, again, is okay, but not the minimum value. And also the maximum position is so-so. It's slightly shifted to the high Q values. And also you have a second maximum here that is not present in the experimental data. Then, what did we do? We added an extra parameter, the ellipticity, and also the rotation, the orientation of the ellipse with regards to the protein. And then we end up with a better model here, which is the orange one, where the second maximum here disappeared. The maximum, the main maximum here is nicely fitted, but still here we have a disagreement with the experimental curve. And so finally, we made steps of 0.1 angstrom instead of 1 angstrom here. As you can see here, the final values have a decimal here. And we ended up with a much better chi value here. And as you can see also from visually from the agreement here. So this is the final model. So this strategy is the strategy with which we recommend to the Memprot users.
So we have a question about Memprot,
Yes.
which is, can Memprot be adapted for a corona of glycans, such as for mucin?
Do you mean a corona of glycans?
Yeah, they made a corona of glycans.
Oh, yes. Then it's a matter of defining the correct density. Yes. So yes. Yes, as you can see, there is nothing here that reminds you a molecule of detergent. We are just speaking of geometrical density here. We are feeling density. So if this density corresponds to glycans, then no problem. Yeah.
All right, thank you. One more question.
Yes.
Would these strategies apply to membrane proteins contained within nanodiscs?
Yes, indeed. This is one of the last implementations that we are commissioning at the time, is adding the nanodisk, because the nanodisk is more complex than the corona. It has also an extra protein density around it. And so it's something that is not yet implemented in Memprot, but the approach would be the same. The approach would be the same, yes. It's just a third. You will need a third density somewhere, because you have the heads of the lipids, you have the tails of the lipids, and around you have a belt of protein in another disk. So we should add this third layer, but it's not already the case. It can be something upon request that could be added. If there is really some interest in it, then it could be a kind of collaboration. Yes, would be interesting.
Okay. Thank you.
So this. So remember, initially we were interested in comparing the solution of truncated aquaporin 0 and full aquaporin 0, but we couldn't make it because we were not able to purify these. But what we did, just for the fun of it, just to check, was to implement in Memprot the PDB file of the truncated protein, even if we were measuring the full aquaporin 0. And see if we could, just by modeling the detergent a different way, could we fit the data? Well, the answer was, I must say, really surprising. We could not fit the data as well as we did with the full model. And this means that despite SAXS can never be modeled by a single model, this approach, how to say it, we couldn't compensate the absence of the external linkers here by modifying the model of the corona. And this is good news, because it somehow it means that when you model the corona, you are not doing anything strange. You are just modeling the corona. And if you don't have the good protein model, if the model that you put inside is the protein part of the model that you put in Memprot is not the correct one, well, you might not end with a good fitting. So retrospectively, it means that if your fitting is not good, it is probably due to the fact that your protein model is not exactly what you have in solution. Maciej Maranovsky spent 1.5 years in improving the speed of Memprot. So initially we used it on a just laptop, and now it is implemented in a cluster with 100 cores. And this is the improvement of speed when you use 20 cores, so it was parallelized, and as you can see from the evolution from one node to 1 core to 20 cores, if it was perfectly parallelized, should give 2.6 trials per second, and effectively it gives 2.16. So we are very close. It's not completely fully parallelized, but it's almost. So the parallelization process, or the way he wrote it, is really very effective. And if you have such a cluster, then I will be happy to provide you the cluster version of Memprot. Because, of course, it's a slow process. Memprot is a slow process. So this is an example of a log file after Memprot run, where it tells you how much time it took to complete it, and it selects the best model, so with the best chi-square, and tells you how many detergent hydrophobic tails would be calculated from it, and how much hydrophilic heads would be calculated from it. And of course, these two numbers should be identical because detergent molecule comes with 1 head and 1 tail. But as they are not, the fact that these two numbers are equal is not a constraint in Memprot, it's an output. Then the proximity of these two values is a kind of check that the model is coherent. So we didn't want to put this equality as a constraint. So to say, we didn't want to put the ratio between the number of electrons to fix the ratio between the number of electrons of the heads and the number of electrons of the tails in the Memprot approach. Instead, we use the output here as a check that the model is correct. So let's come now to the second part of the strategy. So the second step. The second step relies on a completely different program that you can maybe compare to Bunch or SASREF. So it's a rigid body fitting program where your user must implement as an input the structure of the domains whose structures are known. and the program will try to orientate the domains and fit a SAXS curve, but also fit some NMR data like the residual dipole couplings and some ADR score. ADR is something that may come from HSQC NMR experiments that tells that a series of residues are known to be close from one domain, for example, are known to be close to a series of residues of another domain. So this kind of constraints can be put in the DADIMODO. It's very similar to what you can do with SASREF or Bunch or Coral from the ADSAS suite. But the main difference here with the approach of the ADSAS programs is that all the unknown parts are represented as true atoms. So it's not beads here. We represent all the flexible parts as true atoms, true residues. So in this way, we think that we put all the knowledge about the protein, and in particular the stereochemistry constraints that particular residues will put. And some angles between the domains will be forbidden because of this particular stereochemistry. And at each step of the process of the algorithm, we compute the energy from the model using a force field here. So we make sure that at each step, the object that we have is physically viable. It could have this conformation from an energetic point of view. So we have these experimental constraints that we can use in combination or separately, and we also have these computation constraints which is through the energy. And all these constraints are put into a genetic algorithm which will optimize the structure so to fit everything. So the initial version, again, run on a single computer. It was written in 2011 by a postdoc, Guillaume Evrard. And since then, Olga Rudenko, who works at Soleil as a software, she's a mathematician again, and she parallelized it, and we gain, it's 100 times faster now. And in addition to this, we recently our colleagues from Soleil helped us to make a web server that is accessible to external users and through which you can submit your own computations, your own problems, your own subjects. So this is how the web server looks like. As an input, it needs 3 files. So a file with a complete PDB structure of your protein, with all the domains and with also the unknown parts that have to be present. Somehow you have to propose an initial model using modular, for example. Then you also provide, of course, your SAXS data or your experimental data, if you have RDC data or ADR constraints here. In this case, we only used the SAXS curve. And you have to provide a configuration file in which you tell which are the rigid domain in the sequence, from which chain they come, and what is the range of residues that compose a SIGINT rigid domain. This is very similar to what is done in SASREF, and actually we were inspired by the way SASREF proposes the input. So in the case of the protein that I'm going to show, it is made of 3 domains, and one of the domain is made of 2 chains here. a part of the chain A and a part of chain B. And the other domains, rigid domains, are also different parts of chain A here and different parts of chain B. So the program, once you have input the composition of the rigid domains, the program itself will deduce what are the flexible parts, the unknown parts, and will try to mutate them, to change their orientation. So there are two kinds of movement that are allowed throughout the process. The first movement is a 5-psi change in a given residue here, which then result in a single angular change of one domain with respect to another one. And also what we call a double linker axis mutation, which is when you have two domains which are linked by two linkers, then we made a rotation arbitrarily through two residues of two different parts of the linkers. Then one domain is rotated respective to the other one by this axis, and then there is a energy minimization process that will remake the bonds here that were disrupted by this arbitrary geometrical rotation. So the website tells you, also provides some output concerning the history of your submissions, and also tells you if the calculation is finished. And once the calculation is finished, you receive a zip file through an email. And this zip file contains the final PDB files from several runs that are submitted in parallel. When you make one submission, actually 5 calculations are done in parallel, and I will show you why in a few slides. A reminder of the 3 input files and all the log files concerning the run, and in particular for each run, the way the chi-square that you can see in red here is decreased and finally reach a constant value at the end of a given run here. So we started with the initial model gave a very bad chi-square, and as the program goes, the chi-square decreases, and finally we have values which are much better. I forgot to say that here again, DADIMODO uses Chrysol to compute the fitting curve. So this is an example. The example I just described, corresponding to the input file I've just described, where the initial structure in red was the crystal structure. So the people came. This is a collaboration with the Pasteur Institute in Paris, with the DNA gyrase, which is made of 3 domains, and in the crystal structure, the 3 domains make a single globular conformation. But the actual SAXS curve is the black one, and you can see that the initial model in red doesn't fit at all the curve. And so by maintaining the rigidity of the 3 domains, but letting the linkers change or reorient, DADIMODO could provide a model, the green model, where the experimental curve is perfectly fit. But of course, you may be skeptical about this model. What does it mean? So this is the reason why we launch several runs, independent runs in parallel. And you can see that all the final models are different, slightly different. They all give more or less the same kind of agreement. So the chi-square are very low. The fit is very good in all cases. But you can see that the orientation of the model of the domains is not exactly the same for each of these models. And of course, because when you see this, you immediately think, okay, the protein, in fact, is flexible. First, the model that we are showing here, each of the model we are showing is just a representation of an average of the conformations. Okay, it shouldn't be thought as a rigid structure. Nobody will believe it, of course. And not only this, but as you can see, even different averages can be obtained, which will fit the data equally well. And so what can we say then from this output? Well, we can say that very certainly the domains are not making a globular structure around the protein, and certainly the distance, the average distance between the external domains and the central domains is something that is conserved throughout all these models. So, and this is already an information for the structural biologist. So, we applied then this two-step strategy to a more interesting problem, which is described here from a biological point of view, structural biology point of view. So we have this transmembrane protein here from a bacteria, a gram-negative bacteria, so with two membranes here, which is an opportunist pathogen. And the question was when this, so the role of this protein is to capture the iron within a heme here that will be outside the bacterial cell. The heme is carried usually by a hemophore here, and sorry, the heme is captured by the hemophore, and is released inside the Has-R protein with a very, very, very high affinity here. And this is the way the bacteria feeds itself with iron. So one of the questions was, what is this signaling domain role? Is there a role of this signaling domain with respect to the capture of the heme here? Is there a relation? Is there a structural relation? So we started doing SAXS with different constructs. The first construct of known structure is Has-R and Has-A here, but without the signaling domain. And this structure is known. So we computed the corona using Memprot, and then we used this corona model to study the whole construct with the known part and, in addition, the signaling domain that you can see here. So to do this, to do the first step, we had to add an extra. It's not an additional parameter, but it's an extra condition to Memprot that we allowed the membrane to mimic the surface of the protein. And without doing this, we were not able to fit the data. And if you think of it, this is something which makes sense, that the extension of the corona depends on how the protein itself, the transmembrane part of the protein itself, is extended in its own plane. So doing this, we had a nice fit of the curve, and these are the two ways this is now implemented in Memprot. And we could then compare, after using DADIMODO, the structure of the whole. We could propose a model for the conformation of the whole protein, including the signaling domain. And we did this for two initial constructs, one with Has A here and one without Has A here. And the output from this is twice. There are two outputs. First output is that the signaling domain is never close on average, is flexible. It is never close on average to the, is not stuck to the main protein, transmembrane protein part, but it is on average at a certain distance. And this distance increases, the distance is larger when the hemophore transporter here, the Has A, is absent is not linking, is not in complex with the protein. And this was the main structural output from this study, and it was corroborated by electronic microscopy experiment which were done later, where they could also see that comparing these two structures, well, on average, the position of the signaling domain was further away from the transmembrane part in the absence of Has A than in his presence. Okay, so this is the roadmap, which I'm not going to develop too much, but we have a roadmap both from DADIMODO and Memprot. In particular, we would like to make a web server also for Memprot. And this is my last slide, where I show all the people with whom I collaborated doing this work, and in particular my team here at Synchrotron SOLEIL. And I thank you for your attention.